Greater Mount Ararat, Aromatic Cedar Petrified Wood Example Number 1 Beam. Cellular and Molecular Diagnosis and Analysis Results by Ronald Stewart. Copyright 2003-2018, all rights reserved. In this example, we see 3 possible wood beams that were discovered by Ark-Quest team number 2 during the summer of 2015. We are going to do both a cellular and molecular scientific analysis. This video is about those results in detail. Now more as follows. On top is the original first aromatic cedar petrified wood example, and from it is projected an area in the yellow rectangular area about 300 UM or microns. The bottom one is the one we want to concentrate on, where it has a microscopic measurement scale of about 5.0 UM. As we zoom away from the middle 300 UM diameter micrograph, we see that it is very porous, one of the first pieces of evidence for petrified wood. Next, we zoom into the bottom micrograph, which has a microscopic measurement bar in the left bottom corner of about 5 UM. However, the area that is within the oval or circular outline is seen here, going off and on, is the area that we will concentrate on 3 different cells and their nucleosis, and or also known as nuclei. This supplemental material video is based upon a peer-reviewed paper 2018 by Ronald Stewart entitled Greater Mount Ararat, Aromatic Cedar Petrified Wood Example Number 1, Cellular and Molecular Diagnosis and Analysis Results. Although the nucleus images in the accompanying video include these images in Figure 20 of this peer-reviewed paper, these nucleuses or nuclei are not of in the aromatic cedar wood and its nucleus when it was alive. The primary point is that when it comes to cellular molecular application to any cell and how the nucleus functions within the nucleus cell itself, that it still follows the same basic procedures and rudimentary functions as we would see in any form of life, like plants, trees, vegetation, animal, and especially human life forms that are more complex. Therefore, these images and video and images that are made from provides visual, photographic, and videographic evidence of the mitosis of a cell and the function of a nucleus in its action from beginning to end. Therefore, in like manner, just like what you see in the nucleus function in these images and video, it functioned the same way before this piece of wood was petrified. And the cells and nuclei also when they were alive. And this just to give you an example. Therefore, this video is based upon the Creative Commons Attribution Share Alike 4.0 International License by the authors that you see here, Orlov et al., who granted by gracious permission to use the video under this license. Secondly, the authors of this video are neutral and do not sponsor or endorse this video or how may not the images may be used thereof. Thank you for your time. Here are how the 3 nuclei functioned within this petrified piece of wood when it was alive. The nucleus here can be seen, the nuclei inside the nucleus, which is part of the cytoprotoplasm of also the cell. Now, this video shows it starting from beginning to end. Here is the nucleus inside the cell, and of course now we see it change. It's starting to get smaller. It disassembles and then eventually will completely disappear, as we see here next as well. Here we see 3 different cells that have come together, and 1, 2, and 3 are the nuclei that try to save the cell when the damage was too great. Now, the bottom line is that in all living forms alive, the nuclei controls the repair mechanisms of a cell. Therefore, in like manner, when this specific case of these nuclei came together to repair the damage of one cell, the other two nuclei came to help it. However, upon the damage being too great, they all died. So what do we learn here? We learn that the nuclei 1 through 3, once the damage had absorbed the cell, that it spread to the other nuclei and tried to absorb them. However, what they did is they tried to defend themselves but died in the state of ongoing mitosis, as we saw previously in the cell video. We just discussed the top image and what it means. In the bottom left image, A, where the red arrow points to the last image that we just saw in the video. That is the cell and where the nucleus had completely disappeared. However, when looking at this under the AM microscope during the summer of 2015, where the long arrow points to the middle image, this shows disassembled, disorganized nuclear matter. This is the first time in biology or microscopy history that this has ever been viewed here below or at about 1.0 nm nanometers. However, at the bottom of the middle image, when we look at the long red arrow pointing at a right angle that has a yellow outline, it points to an enlargement of an atom that measures about 0.33 nm nanometers, and behind that we can see many examples of the molecular structure of the nuclear matter. And this has never been photographed or filmed with this high resolution image as we see here of an atom before in this particular situation, especially when it shows this inside not a live aromatic cedar tree, but one that over much time has now petrified. The top image shows the 3 cells and the nuclei that came together to repair cell damage but expired. One is the area that we'll be discussing now. The first nucleus, that where the 2 red V-shaped pyramid-like lines are coming from, projects a very important discovery and scientific evidence. The top is a diagram of what we see, what would be the molecular structure of cellulose, which is made up of all plants and trees, etc. Below that is an explanation of the CH group, carbon, hydrogen, etc. cells. In the bottom is the first time ever below the 1 nm nanometer level that there has been a photograph, or in this case micrograph, of a cellulose molecular structure. However, in this case, this is not just an image micrograph of the molecular structure of cellulose at 0.33 nm and or approximately 30 angstroms, but it is a micrograph of aromatic cedar petrified wood cellulose molecular structure at 0.3 nm. And here's the point. This has never been filmed before. And this is important because you would expect it to be in a live aromatic cedar wood, but for it to be discovered inside petrified wood is very, very unusual and extraordinary as well.